Good morning, all of you. Can you hear me?
Yes, ma'am.
So, in the last class, I have mentioned about the glycosylated hemoglobin, but this glycosylated, it should be uttered like it must be uttered glycated hemoglobin. Why? Even if it is glycosylation of the glucose to the protein hemoglobin, okay, hemoglobin polypeptide chain, but this is the like this glycosylation is non-enzymatic, isn't it? So non-enzymatic, okay, without the help of any enzyme, okay, without that there will be spontaneous glycosylation, okay, there will be no catalysis, okay, there will be no catalysis by enzyme, okay, without the help of any enzyme there is no involvement. Please remember, there is a difference between glycated and glycosylated, okay, glycation and glycosylation, okay, the term the glycated, why? Because there is no involvement of any enzyme, okay, okay, attachment of the glucose to the hemoglobin, there will be no involvement, there is a no need for the enzyme, okay, it is a spontaneous process, okay, it's going on automatically, it occur automatically, there is a no help of, there is a no involvement of the enzyme, that's why it is known as and uttered as glycated, okay, not glycosylated, not glycosylated, glycated hemoglobin, okay, because attachment of the glucose to the hemoglobin, okay, hemoglobin polypeptide chain non enzymatically, okay, non enzymatically, without the help of, without the involvement of the any enzyme, without catalysis, this is the spontaneous, okay, so it is known as glycated hemoglobin, okay, please remember, okay, in the exam you will be asked what is the difference between glycation and glycosylation, then you must know this much, only this much knowing is sufficient, because in the glycosylation we need the help of the enzyme, okay, glucosidase something something, okay, okay, glycosylation we need the help of the enzyme, okay, for the glycosylation. For glycation the process is spontaneous, okay, that is a non enzymatic, there is a no involvement of the enzyme, that's why the term glycosylation has been changed to glycation in case of glycated hemoglobin, because whenever glucose attached to the hemoglobin, that is the non enzymatically, without the help of any enzyme, that's all. So in the last class I have told a little bit about the hemoglobin variant, hemoglobin M, okay, met hemoglobin already finished, then sickle cell hemoglobin S, okay, sickle cell hemoglobin also finished, sickle hemoglobin also finished, hemoglobin S, okay, so finish how the this deoxyhemoglobin is in case of hemoglobin S, it forms the fiber like structure, that one also explained in the last class. Today little bit left, okay, today very little bit left, okay, in the thalassemia also I have mentioned little bit, I'll go details for the thalassemia and thalassemia syndrome today, and the and other, this is already I have mentioned, this thalassemia is a quantitative deficiency, not quality, okay, there is a less of, there is a less production, there is less production, okay, not the abnormal production, okay, production is okay, okay, hemoglobin quality is okay, but quantity is less, okay, synthesis is less. There are some other abnormal hemoglobin which is known as the hemoglobin C, hemoglobin D, hemoglobin E, okay, these are all responsible for qualitative not quantitative, quantitative only thalassemia, okay, quantitative hemoglobinopathy, okay, hemoglobin abnormal hemoglobin, okay, quality is abnormal, okay, abnormal hemoglobin, okay, qualitative hemoglobinopathy which results qualitative hemoglobinopathy, okay, quantity is okay but quality is abnormal, okay, abnormal hemoglobin. In case of hemoglobin C in the beta polypeptide chain in the 6 positions of glutamic acid. Normal hemoglobin in the beta chain 6 position always glutamic acid. Okay. In case of hemoglobin C, that is replaced by lysine. In case of hemoglobin S, it is replaced by branch chain amino acid valine. Okay. But in case of hemoglobin C, that glutamic acid 6 position in the beta chain is replaced by lysine. In case of hemoglobin D, that is the Punjab phenotype, that 121 position glutamic acid is replaced by glutamine. Okay, 121 position glutamic acid, it is a acidic amino acid, but glutamine is a amine, okay, dibasic monocarboxylic acid. Glutamine is the dibasic monocarboxylic acid. It has two amino group, okay, amino group containing amino acid. Okay, glutamine. Okay, 121 position normally in the normal hemoglobin in the beta chain, 121 position is occupied by glutamic acid, that is replaced in case of hemoglobin D, then glutamine. Okay, here I have given the formula. Alpha 2 is the adult hemoglobin. Okay, 2 polypeptide chain alpha 2 in case of Punjab D. Okay, does alpha hemoglobin contain 2 alpha and 2 beta chain? Alpha 2 chains are normal. Okay, like adult hemoglobin. But beta chain in 121 position glutamic acid. Okay, it is replaced by glutamine. Okay, glutamine. GLN, glutamine. Okay, this is a short formula, that is the formula GLN, the glutamine. If it is glutamic acid, GLU, okay, it is replaced by glutamine, 121 glutamine. Like that, this formula for hemoglobin C. Alpha chains are normal, like adult hemoglobin. A for adult hemoglobin, not abnormal, adult, normal adult hemoglobin. And beta chain 6 position lysine, okay, beta polypeptide chain 6 position is replaced by lysine. Okay, and hemoglobin E, this hemoglobin E is the most common variant. Okay, we will get in here hemoglobin E. In the northeast we will get most common variant hemoglobin E. Okay, in our clinical biochemistry lab we used to get hemoglobin E, hemoglobin E very frequently. E trait or D disease, this is a very common variant. Okay, hemoglobin E. Here what happened, beta chain 26 position glutamic acid is replaced by lysine. Okay, but in both cases it is glutamic acid replaced by lysine, but position is different. In case of hemoglobin C, 6 position, and in case of hemoglobin E, which is most common variant, this one 26 position glutamic acid is replaced by lysine. Now hemoglobin E, one by one. Okay, this formula you try to remember this formula, then it will be easy. Okay, then it will be easy to write and remember. It will be easy to remember and to write. Now hemoglobin E, it is the second most prevalent hemoglobin variant. Although it is a second most prevalent variant, but we used to get hemoglobin E most frequently here. Okay, in our clinical lab we used to get most frequently hemoglobin E, isn't it? Hemoglobin E is very common. It is the second most prevalent hemoglobin variant. It is due to replacement of beta 26 glutamic acid by lysine. It is primarily seen in Oriental of Southeast Asia, Thailand, Myanmar, Bangladesh, etc. The variant is very prevalent in West Bengal in India. Even in Assam also, in the northeast, in the Assam also, it is very prevalent. Okay, even in the Garu population, I have found hemoglobin E is very prevalent. Okay, from my personal experience, whenever I worked in a private lab, I used to get very frequently hemoglobin E. Okay, hemoglobin blood sample is coming for the hemoglobin typing. I used to get hemoglobin E, that is the E trait or diseases in the Garu population. It is the variant is prevalent in West Bengal in India. Heterozygote. Always remember this heterozygote. Also known as hemoglobin E trait. All the heterozygous is known as trait. Okay, and homozygous. If it is homozygous, it is known as disease. Okay, if it is homozygous, in case of homozygous, hemoglobin E homozygous, more than 90% hemoglobin is hemoglobin E. Okay, don't forget that one. In case of hemoglobin E homozygous, if it is homozygous or E disease, Homozygous also known as E disease. Okay, homozygous or disease, heterozygous or trait. If it is homozygous or disease, more than 90% hemoglobin is hemoglobin E, isn't it? But in case of trait or heterozygous, those people up to 35% hemoglobin is hemoglobin E. That is, you have to write that one. Please write that one. In case of hemoglobin E heterozygous, okay, or E trait, hemoglobin E up to hemoglobin E percentage up to 35, okay, up to 35, 30 to 35, isn't it? In case of homozygous, hemoglobin E homozygous or hemoglobin E disease, okay, homozygous also known as E disease, that hemoglobin E percentage is less, it is more or equal to 90%. If it is E disease, hemoglobin E disease or hemoglobin E homozygous, that hemoglobin E percentage, it is more or equal to 90%. If it is heterozygous or E trait, that hemoglobin E percentage up to 30 to 35%, isn't it? Okay, fine. Hemoglobin C now. Hemoglobin C, in normal hemoglobin A, sixth amino acid in the beta chain is glutamic acid. It is replaced by lysine in hemoglobin C. The presence of hemoglobin C is seen mostly in black race. If it is heterozygous, that is AC. If it is heterozygous, that means in that heterozygous patient, okay, heterozygous person, that the blood hemoglobin contain mixed variety, okay, there some fraction is C and some and little bit adult hemoglobin, okay, adult hemoglobin and abnormal hemoglobin is mixed, okay, in case of heterozygous. But in case of homozygous, more than 90%, almost all the part of the hemoglobin, all the portion of the hemoglobin is abnormal hemoglobin in case of homozygous or disease. In case of heterozygous, it will be mixture of the normal adult hemoglobin and abnormal hemoglobin, okay, whatever hemoglobin it is, okay, hemoglobin C, E, S, okay, D, whatever hemoglobin. In case of trait or heterozygous, the hemoglobin will be the mixture of the normal adult hemoglobin and abnormal hemoglobin in case of heterozygous or trait, okay. Again, that if it is heterozygous, that is the AC mixture, heterozygote, they don't show any clinical asymptomatic, they don't show any clinical manifestation, right? But those are double heterozygous. For double heterozygous also very common, okay. Here in Shillong, we used to get double heterozygous for E beta thalassemia, okay, thalassemia patient having the abnormal hemoglobin, both the quantitative and qualitative deficiency. E beta thalassemia is very common, okay. E beta thalassemia in our clinical lab, E beta thalassemia type of hemoglobinopathy, okay, that is the double heterozygous thalassemia trait and hemoglobin E trait, they are coexist together, okay, they are coexist. This type of double heterozygous is very common, okay. But those who are double heterozygous, but here it is different, but those who are double heterozygous for hemoglobin S and hemoglobin C, it is known as SC, have a moderate disease, okay, moderate disease. These are the double heterozygous. Then homozygote CC have mild to moderate hemolytic anemia, okay, mild to moderate hemolytic anemia. Then hemoglobin C is slower moving than hemoglobin A on electrophoresis at alkaline pH. Even hemoglobin E also mobility is less than the normal hemoglobin while doing the electrophoresis. Obviously, here the valine is the branch chain amino acid, non-charged, and here before in the adult hemoglobin, 26th position is glutamic acid. So glutamic acid movement will be more towards the anode, isn't it? Towards the anode, towards the positive electrode, the movement will be more. So electrophoretic mobility will be more. But in case of hemoglobin E, if it is replaced by lysine, Okay, lysine or hemoglobin S is valine. Okay, that these are non-polar. So electrophoretic, their attraction towards the anode, okay, towards the anode, it will be less, so mobility will be less. Okay, it stop running in between. Okay, whether the normal hemoglobin, it runs towards the anodes, extreme point of the anodes. Okay, like that, the hemoglobin C also moving slower than the hemoglobin A on the electrophoresis, okay, at alkaline pH. Now hemoglobin D. Hemoglobin D does not produce sickling. Hemoglobin D Punjab result from replacement of the beta 121 glutamic acid by glutamine. That hemoglobin D migrates similar to hemoglobin S on electrophoresis. Hemoglobin S and D heterozygous, okay, double heterozygous disease is the severe condition. Okay, if hemoglobin D is coexist along with the hemoglobin S, this double heterozygous is the clinical manifestation will be very severe. Okay, the condition is very severe. Okay, hemoglobin D Punjab is the commonest hemoglobin variant seen in the Punjabi population. Here also electrophoretic mobility is very slow, isn't it? Here, mobility where the normal adult hemoglobin, it tries to cover the extreme point of the anode, and this hemoglobin D during the movement of the electrophoresis, it stops in between. Okay, because this polar amino acid glutamic acid replaced by the non-polar glutamine. Inheritance of hemoglobin variant. Inheritance of the hemoglobin variant. They are inherited as codominant or recessive trait. One beta chain gene is inherited from each parent. Okay, if both parents are heterozygous for S, if both parents are heterozygous for S, suppose here this is this diagram is not for this line. Suppose this parents is heterozygous for hemoglobin C and this parents is heterozygous for hemoglobin S. Okay, hemoglobin S. So there will be chances of the normal offspring up to chances of the normals offspring that is the 25%, isn't it? Then sickle cell trait also, that is chances of sickle cell trait here, sickle cell trait, normal offspring 25%, sickle cell trait, okay, or hemoglobin C trait, there will be 25% and 25%. Here also there will be 25%, here also chances is 25%, but and altogether 50%. Okay, and homozygous, the disease there will be chances of 25%, isn't it, like that? If it is only if the both parents are hemoglobin S heterozygous or hemoglobin S trait, then also chances of the normal offspring 25%, chances of the heterozygous 25, 50%, and chances of the homozygous or disease is 25%, isn't it? But here we have in the figure I have showed the mixture. Okay, mixture. Okay, if it is not blue, it is round or this one like sickle cell, it will be this diagram also same only. Okay, the chances of the offspring of the different variant will be same only, percentage also same only. Okay, because for the heterozygous, okay, the offspring has to get the one abnormal beta chain from either any parents. Okay, the chances of getting that abnormal beta chain of any one of the parents, okay, that's why only the chances possibility of the trait 50%. Got? Now if genotype is SS, okay, if the both beta chain is abnormal SS, all the hemoglobin produce are abnormal. This is a homozygous or S disease. Okay, both the beta chains are abnormal hemoglobin S, isn't it? S chain. Instead of beta chain, it is a S chain, Okay, both the beta chain, glutamic acid 6 position is replaced by valine. Okay, in that case, the clinical manifestations are very prominent. Okay, and condition is known as hemoglobin S disease. Okay, S disease. If one parent is heterozygous for hemoglobin S and another for hemoglobin C, the hemoglobin C or beta thalassemia, 25% chances of that child will be double heterozygous. Okay, double heterozygous. See, this one is double heterozygous. Okay, 25% chance of the double heterozygous. This offspring, this fraction of the this percentage of the offspring is having both hemoglobin C abnormal beta chain and hemoglobin S abnormal beta chain, isn't it? That is percentage is 25%. If it is in place of C, it will be if the parents are thalassemia trait, okay, beta thalassemia trait, then also this 25% chances of the hemoglobin S thalassemia. Okay, hemoglobin S thalassemia trait. Okay, this C is replaced by thalassemia polypeptide chain. Okay, thalassemic beta chain. Okay, now beta thalassemia. Recapitulate what is the thalassemia once again. Thalassemia, in the last class, this definition I have given. The genetic defect known as thalassemia results from partial or total absence of one or more partial absence. There will be, if it is partial absent, the chain is not completely disappear. Okay, only a fraction of the chains are less. Okay, lack of the production, because the lack of the production. Okay, only fraction, only a part of the chain, either alpha or beta, are absent. Okay, not total absent. Okay, so result from partial and total absence of alpha and beta chains of hemoglobin. Over 750 different mutations have been identified, but only 3 are common: either alpha chain thalassemia, that is known as alpha thalassemia, or beta chain beta thalassemia. Okay, according to the involvement of the chain. Okay, a superscript indicate whether the subunit is completely absent. If it is completely absent, alpha chain is completely absent. If alpha 1 is completely absent, alpha 1 0. If alpha 2 is completely absent, alpha 2 0. Okay, if beta 1 is completely absent, beta 1 0. If beta 2 is completely absent, beta, okay, beta 2 0. If both are absent, beta 1 0 plus beta 2 0, like that. Okay, and whether its synthesis, if synthesis is reduced, okay, synthesis is reduced, that is the partial absence, isn't it? Partial absence, a part is not synthesized. Okay, but whatever part is synthesized, that is the normal polypeptide chain, nothing abnormality like other hemoglobin, qualitative hemoglobinopathies, isn't it? Whatever it is synthesized, it is quite normal, but synthesis is less, isn't it? That's why that is the synthesis is partial. Okay, partial synthesis. Okay, due to this partial synthesis, one part of the hemoglobin polypeptide chain is missing, like that. If it is partial synthesis, lack of production is plus. Okay, plus superscript, alpha plus and beta plus, like that. Now beta thalassemia. Beta thalassemia is more common than alpha variety. Beta type is characterized by a by a decrease or absence of synthesis of beta chain. As a compensation, gamma or delta chain synthesis is increased. Okay, body has compensatory mechanism to make up all the loss. So as a compensation, as a result of that, gamma and delta chain synthesis are increased. Okay, in case of alpha thalassemia, they may result from different types of gene deletion is a one type of mutation. Okay, point mutation. Okay, point mutation. Since there are two pairs of alpha genes per cell, a single gene deletion is one chromosome, a single gene deletion in one chromosome, or a pair of gene in the chromosomes does not have much effect on the chain production. isn't it? Since there are two pairs of alpha gene, two pairs, okay, two pairs of alpha gene per cell. So a single gene deletion in the one chromosome or a pair of gene deletion in the chromosome, it does not much affect the chain production. So alpha thalassemia is rarer because alpha chain deficiency is incompatible with life. Now thalassemia syndrome. Thalassemia syndrome. What is thalassemia syndrome? These syndromes are mainly seen in the people of Asian, African and Mediterranean region. All these of thalassemias are characterized by deficiency of the hemoglobin A synthesis. Deficiency of the adult hemoglobin. Usually presentation is anemia. What type of anemia? Histologically microcytic hypochromic. There is a deficiency of the hemoglobin synthesis. As a result of that, the cellular size is very small, microcyte. Okay. So anemia is the microcytic hypochromic or hypochromic microcytic. Okay, microcytic means cellular size is very less as a result of decreased production of the hemoglobin. Hypochromia is there, decreased production of hemoglobin, hypochromia. So anemia type is hypochromic microcytic. Otherwise, number of the RBCs are okay. Okay, but RBCs are hypochromic microcytic. Okay, function will be less because of the defective hemoglobin. In homozygous state, clinical manifestations are severe, hence called thalassemia major. See, here is the difference. There in the abnormal hemoglobinopathy, qualitative hemoglobinopathy like hemoglobin C, hemoglobin E, hemoglobin S, hemoglobin D, those homozygous are known as hemoglobin homozygous. Hemoglobin S homozygous, hemoglobin C homozygous. According to the hemoglobin, it will utter hemoglobin homozygous or disease. Okay, hemoglobin S disease means hemoglobin S homozygous. Hemoglobin E disease means hemoglobin E homozygous, like that. Okay, but here in the thalassemia, if it is homozygous, it's known as major. Okay, major. That means it is thalassemia homozygous. Okay, thalassemia major, like that. Okay, so there will be nucleated RBC in the peripheral circulation. Okay, there will be nucleated RBC in the peripheral circulation, isn't it? Why nucleated RBC in case of thalassemia major? Body cannot synthesize the normal hemoglobin, isn't it? RBC are synthesizing normally, but body cannot synthesize the normal hemoglobin because there will be some defect, genetic defect lies. Okay, genetic defect lies. Okay, that's why the production of the beta chain or alpha chains are deficient or absent. Okay, in case of major. Okay, there deficiency of the beta chain in beta thalassemia, there beta chain deficiency. So as a result, body try to, okay, as a compensatory mechanism, body try to increase the synthesis of the RBC, isn't it? Whenever the defective hemoglobin is there, that RBC is there, but that hemoglobin is not able to carry the oxygen, not able to do its normal function. So as a result there, body tries to increase the synthesis of the RBC. Okay, increase the synthesis of the RBC, increase erythropoiesis, isn't it? To fulfill the body demand. Okay, to fulfill the body oxygen carrying capacity. Okay, fulfill the oxygen, to fulfill the body demand, isn't it? That's why the erythropoiesis will be increased. As a result of increased erythropoiesis, there will be increased nucleated RBC, isn't it? Normoblast. Okay, normocyte, normoblast, like that. Okay, so increase nucleated RBC as a compensatory mechanism. Okay, we will be able to see so many nucleated RBC in the peripheral circulation. That is the main cause. Okay, why nucleated RBCs are more in case of thalassemia major as a result of compensatory mechanism. Because of this thalassemia major, there will be increase the erythropoiesis. Heterozygous condition, heterozygous is known as minor. Okay, Here in case of thalassemia, homozygous known as major and heterozygous known as minor. Heterozygous condition here, thalassemia major, clinical sign symptoms are very negligible, okay, almost like normal person, okay, and most of the time this thalassemia trait, the minor also known as thalassemia trait, but major is homozygous always known as major. This minor, it is a thalassemia trait, it coexists along with the hemoglobin C, hemoglobin E, hemoglobin S, like that, okay, hemoglobin D, like that, it coexists with the qualitative hemoglobinopathy. The synthesis of the unaffected chains occurs at the normal rate, okay, unaffected chain, because 2 alpha 2 beta, no, those chains are unaffected, their synthesis are quite normal, okay. Since they do not have complementary chain to bind, although this normal chain synthesis occurs quite normal manner, but they don't have the complementary chain to bind, okay, because this complementary chain production is deficient or there will be no production, total absent. So they aggregates, those chains are normally synthesized, they don't get their partner, okay, their complementary chain for binding. As a result of that, they aggregates and precipitate within the cell and resulting the inclusion body of this precipitate. Inclusion bodies inclusion bodies lead to membrane damage and destruction of the red cells, okay. These inclusion bodies results in the destruction of the RBC membrane. As a result of that, RBC lifespan will be less, destruction of the RBC. That is the consequences, thalassemia syndrome. The coexistence of hemoglobin S and beta thalassemia trait is fairly common. Okay, although here in the Shillong, I used to get hemoglobin E beta thalassemia. Okay, hemoglobin E heterozygous is coexist with thalassemia minor, okay, or thalassemia trait, isn't it? Thalassemia minor or thalassemia trait, okay. But but overall coexistence of the of hemoglobin S and beta thalassemia is common, okay. But although it is very common, but here I find coexistence of the thalassemia trait along with hemoglobin E trait, okay. Homozygous beta thalassemia is characterized by severe anemia, hypersplenism and hepatosplenomegaly. Okay, both the enlargement of the spleen and liver. Okay, and there will be increased cellular multiplication of the splenic cell. Okay, inside the spleen there will be increased cellular multiplication and severe anemia. Okay, the marrow in the skull bone expand, producing the marrow of the skull bones expand, producing the hair on end appearance described in the X-ray. In case of thalassemia major homozygous beta thalassemia. Beta thalassemia homozygous, it is known as thalassemia major, don't forget, major. Okay, that is in the X-ray, there will be hair in on appearance, expansion of the skull bone. Okay, there will be expansion of the skull bone. Repeated transfusion is only available treatment. So in that case, we have to do the repeated blood transfusion. Okay, this may lead to iron overload sometimes, and splenectomy may also to minimize the anemia. Okay, splenectomy you can do to minimize the anemia. Marrow transplantation has been successfully tried in a few cases. Okay, bone marrow transplantation also, that is also successful, only few cases, not that successful, only few cases it is successful. Now hemoglobin LEPOR, this is composed of alpha chain and delta chain, delta beta, it is composed of 2 alpha chains, okay, 2 alpha chains and 2 delta beta chimeric chain. Okay, 2 delta beta chimeric chain. The N terminal made up of delta chain and C terminal is made up of beta chain. Okay, see, in case of hemoglobin leper there are four chains, two alpha chains. Okay, that is normal. Okay, but two chains, remaining two chains are not even beta also, not completely beta also, and not completely delta also. These two chains are chimeric chains. Okay, that is known as delta beta chain. And why? The N terminal of that delta beta chain is composed by delta chain and C terminal is made up of beta chain, that like that. Okay, in case of hemoglobin leper. Non-homozygous crossing over of the chromosomes is causes for this chimera. Okay, non-homologous crossing over. Non-homologous crossing over. There is an abnormality of the crossing over. Okay, over the chromosome is the causes of this chimera. That's all about this hemoglobin variant. Okay, please try to study. Okay, thank you.
Thank you, ma'am.